This is the Anthrobiology Podcast, hosted by me, Gabby LaPera. Every 2 weeks, I interview a biological anthropologist about what makes us, us. Hello, everyone. Welcome to another episode of the Anthrobiology Podcast. Joining me remotely, I have Dr. Maria Nieves-Colón, and I have her grad student, Jamie Zolik. Hello. Welcome, both of you. How are you?
Hi. Thank you. I'm doing good.
Yeah. Hi, Gabby. Thanks for having us.
Yeah, of course. It's really nice of you guys to join me. I have you both here from the University of Minnesota, and I actually invited you both because I went to a presentation that Jamie did at the American Association for Biological Anthropologists conference in Baltimore this year, and it was amazing. It was honestly like the coolest thing I've ever seen in terms of community engagement. I'm sorry, I'm just like, it was so good. Well, I'm not telling you guys why. It was so good. It was some of the most interesting work and most complete work that I've seen in community engagement since I've been in the field. And I really wanted to have you guys on to talk about your research and kind of your research ethos as well. So yeah, let's just go ahead and get into it. I guess the first thing that I want to ask you guys about is kind of the main thrust of your lab's research, which is mostly about ancient DNA. And you're at this particular site in this town in Peru called San Luis de Cañete, which is like south of Lima. I looked it up and it's near the coast. It looks really warm. Every single picture I've seen of people, everyone looks like a little hot. Can you tell me a little bit about like what prompted the start of this project?
Yes. So this project began with the community of La Quebrada wanting to understand the history of their ancestors. And so in the town, there used to be a sugar plantation. And underneath the main plaza of this town today is a historical burial that contains the enslaved African and Afro-descendant individuals that were forced to work on these sugar plantations. And we know from historical records that these individuals were buried at this site between the 18th and 19th century. And so the whole project began with a community wanting to understand more about these individuals because there's not many historical records that say anything about the lives of these people. We have some historical records that say baptismal names, and so some of the names in these records actually correlate to community members who are alive today. And so we have a connection from these records of these people to the community, but the community really wanted to explore finding more information about these individuals. And one way that they've done this is through previous archaeological excavations conducted. This is prior to us becoming collaborators. The excavations uncovered 245 individuals, and they were performed by Dr. Claire Mass and Jose Santa Cruz. And we came in as collaborators. The community reached out to us to further analyze these ancestral remains using ancient DNA. And so we came as a collaborator, I believe, in 2021.
Perfect. So what was the previous analysis more like osteological, or like what kind of stuff did they do before? Because ancient DNA is like typically kind of like a little bit farther down the path. Like someone has to be like, let's find money and do stuff.
Yeah, that's correct. They did archaeological, bioarchaeology. They tried to analyze any cultural material left behind in the burial site. So they found through this analysis that majority, more than 60% of the individuals within this cemetery were individuals younger than 21 years of age.
Wow.
A lot of individuals in the cemetery are subadults.
Yeah.
Could I add to that?
Yeah, of course.
Yeah. I'd also add that the previous osteological analyses suggest that a lot of the individuals who are interned in the cemetery don't have markers of chronic illness. So if you think of a cemetery like a medieval cemetery or like a monastery or places where people lived long lives and may have had illnesses that accompanied them through a lot of their lives, a lot of those individuals often have markers of illnesses like leprosy or tuberculosis or even arthritis and things like this. In the cemetery that we're working with, we actually don't see a lot of those indicators, but we do see indicators of non-specific stressors. So things like poor nutrition can lead to markers in our bones. And we do see some of those, especially in children, some of the subadults. And we also see markers of intense physical activity, which is really important because it suggests to us that very young individuals are participating in the economic life of this plantation. And so enslavement is impacting what they're doing with their bodies from a very early age. But when we think about slavery, often we think of adult men working in these plantations. And we do see that, right? We do have adult men there, but we're also seeing markers of activity in women, in young girls, and in young boys that suggest that they're really participating in the labor structures of this hacienda in ways that perhaps from the historical record we were really not able to grasp. And a lot of that analysis is done by Dr. Mass, our collaborator.
Yeah, that's really cool. I think that is a really important thing to remember. I mentioned on the show a few times that like academically, I came up in the colonial tidewater in Virginia. So we also had quite a few like enslaved populations and things that we looked at. And one of the wildest things I ever saw was Schmorl's nodes on someone who was like 15 or 16. And these are like huge depressions in the vertebrae from basically the, what is it called? The little disc that, yes, the vertebral disc bulging out. I was like, what is the disc called? It is called a disc bulging out from between and everything grinding together. And to see that in like a 16, 17, 18-year-old is nuts. But that's just like the degree of physical labor that they were experiencing. This is something that you typically see in like an 80-year-old.
Right. So we do see, and this is not an analysis that we have led. It was led by Dr. Mass, but there are signatures of stress to the column, stress to the long bones, activity markers, carrying high loads, repetitive movements, right? Those lead to something called an enthesopathy, right? So like a hardening of tissue that leads to hardening of bone. And we also see, importantly, a lot of enamel hypoplasia, which happens when you experience stressors early in your life while your teeth are still forming. And we see that a lot and sometimes multiple times in some of the individuals in our study, unfortunately, very early on in their life.
Yeah. And for listeners, enamel hypoplasia is basically like lines on the enamel that if your nutrition varies as your teeth are forming, it'll like cause greater or lesser deposits of enamel. And so you get like these really interesting lines. If you Google it, you'll see a lot of really excellent pictures. Okay, but this is your collaborators. They did all this work and they did a ton, it sounds like. And then they were like, you know what we really need is we need someone to do the DNA. And that's when y'all got called in, right?
Yes. And I just want to add one thing that our collaborators did because it really relates to why we're here, which is they identified signatures of intentional dental modification in some of the individuals from the cemetery. So in some human cultures, actually quite a lot of human cultures, there is a tradition of modifying teeth for aesthetic reasons or for social identity reasons. And we know that in many historical communities in Africa, this was also a tradition. And I just want to emphasize for the listeners that this is like something that requires specialized knowledge, right? This isn't something that like your average person could do. These are basically dentists who are doing this. And when the transatlantic slave trade happened, people who had these modifications from different areas of Africa were brought to the Americas, and they came with these modifications in their mouths, right? So when we identify people in cemeteries that have these modifications, we have a pretty good indication that some of these individuals are the people who were kidnapped and forcibly transported from Africa. But we cannot just use this marker to tell us from where specifically in Africa. We can do some comparative analyses, but we need a bit more information. And so that is part of the reason why the ancient DNA component of the project began, because there was a lot of interest in understanding where within Africa did some of these ancestors come from, what was their genetic diversity, what were their biological relationships to each other and to the descendant community, and then what was their health like, right? And how does all of this help us understand the life of this community today, their relationship to the past, and the legacy of these ancestors and the broader sort of transatlantic slave trade in Peru and Latin America?
Yeah, I have so many thoughts. One of the things I do want to say to listeners is if you're interested in tooth modifications, which I think everyone should be because they are really heckin' cool. If you Google tooth modification, Africa, like 1900, 1800, like some sort of like date range, you should get some images on the internet. Just in case you can't, I'll put some up on the website. We were really lucky at the Smithsonian. We had some like really amazing tooth modification, skulls with tooth modifications, including some with inset jewels, which was amazing. Super neat. And then the second thing I actually wanted to ask you guys, because I don't know how familiar people are with the geography of Latin America, but Peru is on it's on the Pacific side of Latin America. And we're talking about the Atlantic slave trade. Do you have any idea of how people got to Peru?
Yeah. Actually, historians have looked at this in a lot of detail. Most of the people who were enslaved in the Pacific coast were actually not coming directly from Africa, but instead were probably being imported into Atlantic ports like Cartagena, Colombia, or in Brazil or in the Caribbean, and then making a really long land route, right? Going in caravans or maybe through river boats to the Pacific coast. Now, there are other people who were directly imported through the port of Callao, which is where the airport in Lima is right now. It's that area there. But often these people were then put on boats in like Bahia and then moved along the coast of South America to Peru. So while there are some people that are coming directly from Africa, likely they are arriving into the Americas and then going through a long period or a long journey into these other places. So if you can imagine what that must have been like and how that sort of sets up your health and also the separation from your community and your family, like, right, this is a very intense journey to be making.
Yeah.
And there's a lot of knowledge that is lost along the way of that.
Yeah, definitely. Yeah, definitely. And it's not like it's exactly like a bustling city center today. Like this is like a rural kind of hard to get to place even today with like modern conveniences like cars.
Yeah, I would say today it takes about 2 hours from Lima to arrive. And while it is a rural place, it was always connected to the metropolis. And this matters a lot for our study because, as we'll discuss in a minute, we're starting to see that connectivity in the genome. And also because we recently discovered that there are some historical records that talk about the peoples who were in these haciendas, because sometimes they would go to Lima to either receive medical care or to complain to the authorities because they had some sort of complaint against the owners of the plantations. And so we do have some records because they were close enough to Lima that they could make it. It probably would have taken more than 2 hours. Today there's a highway, but you know.
Yeah, that's super cool. So I think let's go ahead and like talk about your component, which is ancient DNA analysis. I guess first question is what is ancient DNA? What does that mean?
Ancient DNA is essentially DNA that is in a living organism that is no longer living. So the moment that an organism dies and is no longer able to keep cells alive, the DNA begins to degrade. And it degrades in a specific way that is not so, I wouldn't say predictable, because we cannot really kind of, it can degrade faster in certain environments compared to others, but there is a pattern of DNA fragments. So what it used to be a long fragment, now it's very tiny. The bases at the exposed ends of DNA also change. And so these are ways that we also can verify DNA is ancient and not from modern contamination.
Yeah, totally. So I think to me, in my head, like the thing that distinguishes ancient DNA from like regular DNA testing is that regular DNA testing, it's like you have so much DNA that you can test. And then ancient DNA, it's all fragmented, it's older, it's, as you mentioned, it's degraded, there could be other contaminants. So like you have a smaller amount. Earlier this year, I talked to Dr. Henry Ehrlich, who helped invent PCR tests, which are super important for ancient DNA because of this degradation, right? Because, sorry, I'm taking your thunder. Tell me all about, tell me how do you go about, you have this ancient DNA and it's all degraded. How the heck do you examine it?
Yes, I mean, so this is a large conversation also, because so when you're dealing with modern DNA, you can take it through non-invasive means. I mean, saliva has a lot of DNA, hair can have DNA, you can go back and get more of it. When you're dealing with ancient DNA, oftentimes the individual doesn't have any soft tissue left. And so you have bones primarily that you can extract DNA from. And once it's a destructive process, so you have to grind up the bone, you have to extract the DNA, and there's not much in there. This is a big part of our project because we have to be very intentional about what type of sampling we take with ancient DNA, because you essentially destroy it. So this is a conversation we have with our community of what type of sampling that they were comfortable with these ancestors. We can come back to that later.
Could I add something, Jamie?
Yeah.
Yeah, I'd just like to mention also, if you'll remember, we were talking about how the skeletal remains of the ancestors sometimes have markers that are informative about their social identities in life or their illnesses that they had. So we have to be very cautious not to sample those elements because they will be destroyed forever. Or if we do sample them, to really thoroughly document them, because then we would be losing information that would be helpful for actually making sense of the molecules that we might get. So like Jamie said, this has really been a conversation just starting from sampling before we even do anything. We have to think really critically about what part of the skeleton will have the most DNA preservation, but then also balance that against archaeological or historical importance or even cultural importance of these elements.
Totally.
Yes. And there's also other methods that are being developed to lessen the damaging bones and having to destroy them. There's a sampling of taking DNA from soil. So as an individual decomposes, the idea is that the DNA can be captured in the soil. There's also other methods being produced of dipping, I believe, a bone sample and getting the DNA, human DNA, off of that way. But these are developing, and we had a lot of conversations about what kind of questions we can ask with these different types of sampling methods. And so for our project specifically, we all decided on sampling a single tooth from each individual and sampling the inside dentin layer of that. And so that's where we checked our DNA from.
So is that about what you would take? You would dissolve an entire tooth?
Not us. So for our project, human DNA can be preserved in the inner dentin pulp layer of a tooth. For us to get that, there's also different methods of how to dissect a tooth, how to create the tooth into bone powder. We didn't want to destroy the whole tooth. That was an option of just like powderizing the whole tooth. So what we did is we sectioned the tooth and used a Dremel to kind of just carve out the minimum amount of bone powder we needed for our project. We have a majority of each individual's tooth still remaining within our facilities, either to use for additional questions that us or the community want to investigate, or possibly return their remains back to the ancestors once the project is complete.
Super cool. Yeah, I was like, I didn't want people thinking you guys were like taking like half a leg bone or something.
No.
Just like
I would just add that part of the reason why we decided to sample dental pulp is because it's where your blood vessels would have been during your lifetime, right? That's the part that when you go to the dentist, they have to numb it because they're going to do something and they don't want you to be in pain, right? Your nerves, your blood vessels are there. So it gives us DNA from the person, but it may also have DNA from the microbes that are in the bloodstream of this person. And that might be commensal microbes, but it might also be ancient pathogens that could have circulated. And so we are getting a lot of information from one sample versus other sampling methods or elements might only give you information about either the host or the pathogens. And so here we're trying to balance both.
Yeah, I never really thought about that, but I guess like for listeners, like when you have your ancient DNA, like you put it through this process called amplification, where you basically take like little bits and then you make way more of the little bits.
Yes.
But I hadn't really thought about this till I talked to Dr. Ehrlich earlier this year. You can't really choose what you're amplifying. You're just going to amplify everything that's in the sample.
Yes. And a majority of it, probably 99% of it, is not human, or not even ancient. So another question that I have is based off of the idea that we're sampling an area where previous blood vessels existed. And so if there is a commensal or pathogenic microbe within the system, there is maybe a possibility that we could also get that DNA. And that's even a tinier amount than the human DNA that we have the potential to possibly capture.
That's so cool. That's so cool.
Yeah. And you're right, Gabby, that we cannot select when we extract DNA what just a human DNA. We get everything. But there are laboratory methods that we can do later on downstream to try to fish out those little pieces of human DNA or ancient pathogen DNA. And they are called enrichment or capture methods. where we make baits. Like we literally go fishing in the sea of DNA fragments that we have, and we make these little baits that are going to stick to just the species of interest. So if we're trying to get human, we have baits that are complementary to human DNA, and then we fish out most of the human DNA and we try to get rid of the contamination. It's not perfect, but it's one of the best tools that we have to be able to just get high resolution data from the genomes of these ancient people or their microbes.
This is super cool. So I recall from your talk that you both worked in San Luis, and then you also you did all of this actual DNA work in your lab, because the lab has to have like they're very complicated to set up, and it's not easy to ship all your stuff down to Peru. So I guess, do you guys want to talk about your lab, or would you like to talk about San Luis? Or both, I guess. Which one do you want to start with?
Yeah. Why don't, Jamie, do you want to talk about the lab?
Yes, I'll talk about the lab. Yes. So in order to do ancient DNA, you have to have a specialized lab in order to do it. The lab specifically, we call it like a clean lab. Everything is washed down with bleach and water and gets UV treated every night to kill any modern DNA contamination that might be in the lab. Because when we bring these samples from these individuals to a lab to start the extraction process, we do not want to introduce additional modern DNA contamination than maybe what is already present in the sample. So the lab that we work in is, like I said, gets washed with bleach constantly. It gets UV treated every day. And it also is pressurized in a way that dust and particles don't float freely between the different rooms. And so it is quite difficult to do this work if you don't have access to this kind of laboratory facility. And so when we did this project, we did communicate that the sampling and the extraction and everything would have to be done here in Minnesota. That also led to a lot of conversation of how can we keep the community involved with what's happening to their ancestral remains during this process that we're very far from them. And so we came up with a lot of different ideas of how to get them involved and see what we're doing while we're doing it.
Yeah, tell me more about that.
Okay, cool. So a little bit of background. This project began the same year I began my graduate school program. And so a lot of the conversations that were happening at the start of this was mainly between Maria and the community, and me and my other collaborator, Laura, who's also a student, were brought into the conversation. And so one of the ideas is that while we're sampling, while we're doing all the laboratory methods, why don't we have a GoPro with us and record everything and create a video that the community can see exactly what we're doing from start to, because we can explain it verbally, we can show diagrams, but it's not the same as, seeing exactly what's being done. We got that inspiration from a previous project that did that with their community as well.
Yeah, so the Anson Street Burial Ground Project, which is led by Dr. Raquel Fleskes and Dr. Theodore Schur, they also did a GoPro project with the community that they're working with. And so we saw that and we were like, hey, maybe this would be a good idea. And we kind of said it jokingly. And then the community members were like, yes, do this. And we were like, oh no. And so Jamie and Laura, who is the other PhD student who's in the project, really did all the hard work of like figuring out how do you wear a full body suit to, PPE, personal protective equipment, with a GoPro on your head, and then film and then edit a video that is understandable. And I think that this is really helpful in that it helps us build trust with community members, because when we say we're going to do a certain process, they can understand better what that process is. And it's not so black boxy, or at least we try for it not to be so black boxy. And hopefully that means that we engender some goodwill so that in the future, if we want to do additional research, people, first of all, understand what that is. And secondly, have a really good informed background in order to say yes or no.
Yeah, totally. I think that that's something that I have been glad to see happen in anthropology more and more over time, which is that for so long in anthropology, people have just been like, I'm going to come and I'm going to study this regardless of whether or not you want me to. So it is like, it's it's very different from what it was like even 15 years ago. And it's like, it's great to see it. It makes me very happy. Okay. So you have done all this. Have you had any results yet that you can share?
Do you want me to talk about that, Jamie?
Yeah.
Yeah. So we, like Jamie said, we started the project in 2021. This was peak pandemic, right? So it took us a little bit, but now we have finished the first phase of our analyses. We have sampled 61 of the ancestors from the cemetery. And of those 61 ancestors, so far we have recovered mitochondrial DNA, which maybe your listeners are familiar, right? Mitochondria, the powerhouse of the cell. We need lots of mitochondria in order to have energy. So there are so many mitochondria in our cells that it preserves at much higher proportions than the rest of our genome when we're talking about ancient samples. So you can have between 100 to 10,000 mitochondria in each cell, but you only have one copy of the nuclear genome, which is the genome that you get from mom and dad. So much easier to recover mitochondrial DNA than nuclear DNA. And mitochondrial DNA is also really interesting because it's inherited clonally. So I have the same mitochondrial DNA that my mother had, and my mother, the same one that my grandmother had, and so on and so forth. It's like a telescope. It allows us to look back in time at the maternal ancestors of humans, right? So this is where the most of our analyses are right now. It's the first sort of phase of the Ancient DNA Project. And we so far are seeing that the ancestors in the cemetery actually had maternal ancestors that came from different places in the world. So a big majority of the enslaved population in the sugar plantation, which was called Hacienda La Quebrada, had ancestors that came from Africa. We see that they have maternal ancestors. And actually, those maternal ancestors probably come from all over Africa because we see a very high diversity of mitochondrial lineages in these ancestors. So in other words, we don't see everybody having the same mitochondrial lineage. Instead, what we see is most people actually have different mitochondrial lineages that today are found all over the African continent. And that tells you something about how the transatlantic slave trade was really forcibly taking people from really all over across the hundreds of years that this happened. Now, in addition to that, we also see some individuals that have maternal ancestries from the Americas, likely suggesting that some of the people who were enslaved had maternal ancestors that were either indigenous to Peru or mestizo, right? Mixture between Europe and the Americas. And then we have one individual that has a Eurasian lineage. So it's a mitochondrial lineage that comes from somewhere in either Europe or North Africa. We can't quite tell from where, but this is the minority. Most of what we see are people with African maternal ancestries or indigenous American maternal ancestries. And that is really interesting because it tells us that the mixture that we see in present day Peruvian and Afro-Peruvian communities today was already present in the 18th century when Hacienda La Quebrada was operational and when some of these ancestors died, right? So many of these ancestors had an unmixed genetic background. And now as we start looking into the rest of the genome, the Y chromosomes, which are inherited from the fathers, and then the rest of the nuclear genome, we'll now be able to get a much bigger picture of what are the ancestries of these individuals. And can I just add one thing?
Yeah, of course.
I want to say a humbling moment for me, which is that when we first have these results and we went back to Peru to present them to the community, the first year we went back, I was kind of embarrassed to present just a subset of mitochondrial DNA, right? Because if you think about it, we have 22 chromosomes plus XX and XY, right? That's just such a small part of our genome. And I was like, oh my gosh, we haven't made as much progress as I wanted to. And then we got to Peru and we presented the results to the community, and they could care less about how much data, right? They were so interested because they told us that something they wanted to know more about was the role of mothers and grandmothers in their history. Because, unfortunately, the way we write history often privileges the perspectives of men, and there was nothing written about their mothers and grandmothers, yet they had oral histories of mothers and grandmothers passing on Afro-descendant traditions. And so this was something they were really, really interested in. And I mention this because, we're doing this project that has a very big component of community engagement, and that requires us to listen sometimes and kind of refocus the priorities of our analyses. So mitochondrial DNA analysis is now a much bigger part of the project than I think initially we had thought it would be, because it responds to that interest that the community members had.
No, that's really cool. I was wondering, two questions. One, did you find any people who shared maternal lineages in the group? And then the second question is, are you working with the modern population to sample their DNA to see if they are related to the people that are in the cemetery? So
I can answer the first question. We do today have a couple individuals that do share the exact, like, say, mitochondrial DNA. Now, whether or not that means that they are related through their maternal line, like first generation or back, we don't know at this time. But only a few people share it. So majority do not.
Interesting.
Yeah, it is really, really interesting. One of the questions I want to actually investigate now that we have preliminary data is to do autosomal capture. So capture just the autosomal DNA of these individuals, amplify that, and see if I can investigate kinship relationships between these individuals. For your second question, yes, we are having another phase of the project of analyzing the modern DNA of the community members that are there today. We had 55 participants in this study. Please correct me, Maria, but I'm going to say it this way. Our intention for this project is not to identify who is and who is not directly descendants of the individuals that we are analyzing in the ancient DNA, because one, we can't analyze all of the individuals in the cemetery. Again, there was 245 individuals. We're only analyzing 61, mainly because of limitations of individuals that had teeth available to us that didn't have pathologies or mold. And so we were limited to that. We don't want the conversation to be, again, like who is and who isn't. We know people in the community today. They know that they are the descendants of these individuals from the baptismal names through the last names. What we want to do with this modern DNA project is basically very similar to what we're doing with the ancient DNA. What is the population history of the community? What is their story? And there's a lot that's also happened between the individuals in the cemetery and today. We've interviewed all these individuals that volunteered for this project, and it is really, really cool how much different family history that everyone has. And I think that's the point of the modern DNA portion, or one component of it, and to recognize and celebrate that story.
Yeah, totally.
Yeah, I actually agree. I think you expressed that perfectly. I would also add that the community themselves are interested in looking at how the genetic diversity has changed over time, right? And with the modern DNA sample, we can really kind of create a timeline of how San Luis, which is the district that we are working in, and La Quebrada specifically, have shifted over time since the 18th century to today. And that also allows us to answer some questions that are basic research, but could have some insights into health in the future, right? So we can look at how the genetic diversity of genes involved in immunity and other traits have shifted over time as these communities have been resilient to the challenges they've encountered since the colonial period to today. And so this is research that our other graduate student, Laura, is working on with us, where we are investigating not just mitochondrial DNA, but also some areas of the nuclear genome, or we hope to, once we get enough DNA, to look at how this has changed and how that might be impacting the community today.
This sounds like an amazing ongoing project, and I'm really excited to hear more about it in the future. Like, I'm definitely going to keep up with you guys and let my podcast listeners keep up with what your discoveries are. Is there anything about your research in particular that I haven't asked you about that you'd like to talk about?
Yeah, I think I really want to talk a little bit more about the community engagement that we're doing. So the first year that we started this in 2021, we were unable to visit the community in person, primarily because we're coming out of the tail end of COVID. So it wasn't until the second year that we were able to visit the community in person. And so Maria and I both went down for, I think, 2 weeks in the month of June, which was also the month of Afro-Peruvian history and celebration. And like Maria mentioned before, we didn't know what exactly to expect when we got there. We made all these plans of how we were going to present this information. I talked a little bit, I think, about this in my presentation previously, but we would make a PowerPoint, and then we would spend 3 days in the hotel in Peru fixing the PowerPoint. And we factor that now into our plan when we visit, because when we land, we hear all sorts of different information and interests that the community has and questions. And we want to like alter our way we're presenting to answer those. I also did not speak or understand Spanish prior to me beginning this project. I started learning the year I got involved in this project, and by year 2, I still couldn't communicate. And so I knew that. And so I knew that I wouldn't be able to hear and listen as well as Maria, who that's Maria's first language is Spanish. I kind of have to go off of body language and just looks, which I'm fine with and I'm fairly good at. But the reason I'm saying that, one, is if anyone is looking to get involved in a project, I really want to say, like, learn the language, and it's okay to do it little by little as long as you just keep going. When we did the presentation, I focused mainly on creating animations, creating nonverbal cues to the information that we are presenting. I did not want to leave out information about how DNA is extracted. What can we say? What can we not? But I also didn't want it to be confusing. I wanted it to be accessible and useful. And so I spent a lot of time in the animations. And I think Maria can agree, people understood what we were saying. They asked questions. It was absorbed. So that was a really good feeling. Also, I attempted to say my portion of the project in Spanish. Again, I didn't know how to say it. I had it written down on my cue card. I practice. They know. They really appreciated, I believe, my attempt, our communities in particular. So that was nice.
That's one thing I will say about Spanish speakers is that they are thrilled when anyone tries. Like,
this is true. I think they paid more attention when Jamie was talking than when I was talking, because they were like, "Oh, Jamie's doing an actual effort." You know? Yeah. And I just wanted to add to what Jamie said. Sometimes, we as researchers, as women or minoritized people in science, we might doubt ourselves, right? We might think, "Oh my gosh, I'm not qualified to do X or Y," and we get imposter syndrome. But actually, sometimes those differences make it so that you're actually really well positioned to solve this problem. Because I will say that Jamie's animation work and the visuals that she has created are so clear that I now sometimes use them teaching with proper attribution. Of course, you must always cite your sources. And people are always like, "Wow, that was a very clear image." And so from, necessity, now we have these tools that are making our engagement much more efficient and are also enabling us to talk to all kinds of audiences about the work that we do. So that's a win, right?
Totally. No, the design component is amazing. I had no idea that you were doing that, because that's literally my day job. I write. I'm a UX writer for a fintech startup, and literally all day I sit around with designers and we talk about, like, how do we make this as clear as possible to people who may not speak English? Like, how do you get them to take the next action? How do you get them to understand what's going on? And you did that for ancient DNA, which is wildly impressive.
I love DNA so much. I want other people to at least know about it, because I believe other people will love it too, or if they are able to have the access to understand it.
Yeah.
And things like that.
Totally.
Other things that we made, because again, we are only able to visit once a year, is to leave something physical behind. And so that is how we developed the idea to create physical brochures that we leave. I also want to plug in the fact that a lot of the different community engagement strategies we do are in response to what the community says they want us to do. So for example, I talked about how we do presentations in front of students of all different grade levels. And that is mainly because a community member will call us up and say, "Can you come here today and do your presentation in front of them?" And we say, yes. We've learned to keep a USB drive on us at all times, because that can happen in an instant. And we have to be ready at all times to do the presentations. And so, but I think that it's not just us doing this work. There's no manual of what we should do, how we should do it. It is a conversation. And I think we're just trying our best, all of us in the lab, to just live up to what the community hopes and dreams are for this project and what we can do.
That's a great point, Jamie. These strategies work really well in this context, they may or may not work as well in other contexts. And people, for example, working in the United States or in China or in Mongolia might have different strategies depending on what the needs are of the places that they're in. And I'd also like to mention that much of this work is co-designed with our collaborator, the archaeologist, Jose Luis Santa Cruz. He is himself a community member and also an archaeology professional. And so he really bridges both worlds and helps us translate some of the technical aspects in ways that, he may anticipate the community might want to know more about or less about. And so it's really through that collaboration, through different disciplines, but also people from different backgrounds, that we're able to do a project that is, we hope understandable to people that have different levels of contact with, science and history and education and such.
Yeah, that was beautifully said by both of you. One thing that I wanted to kind of note to listeners is, I think that probably everyone catch this, but Jamie, you are Dr. Maria Nieves-Colón's advisee. You're her student. And I think that this is such like a kind and wonderful advisor relationship. It's so cool to see. Because like one of the things that we talk about on the show a lot is like choosing the right advisor for grad school, because that can really make or break your experience. And like it is difficult. And I have people write to me all the time asking like, how do you find someone who will care for you and not like work you to death or like exile you to Siberia because they didn't like something you said? You know what I mean? Like, so it is like really refreshing to see such like a wonderful, strong relationship between two people.
We just exile people into the clean lab when they don't behave. No, just kidding. Thank you.
Oh my gosh. But yeah, so that is really wonderful. Jamie, what was your grad school application process like? Like, how did you go about picking schools? How did you go about picking advisors?
Yes. So my background, I did my undergraduate at the University of South Florida in cellular molecular biology. At that time, I was really interested in DNA, particularly epigenetics. And from the first year, I was really involved with research in a lot of different research labs. At the end of my time there, I worked with plants, I worked with house sparrows, and I didn't want to do that anymore. I really was interested in evolution, and I thought the best way to do that is to have understanding evolution, is to have access to information about DNA from the past, so ancient DNA. I knew that about myself at my senior year of college, but I didn't really know exactly what I was doing. And so I took that opportunity to just do a master's, and I continued my master's there for 2 more years at the University of South Florida in archaeology and applied anthropology at the time. During that time, that's when COVID happened. And so I think finding a graduate program was different, and it's probably going to keep becoming different as time goes on. I had a lot of different types of advisors, so maybe not one that was in charge of me with my academics, but in charge of me in the lab and things like that, and guiding me and being a mentor. So I had a lot of experience of what I liked, what I was looking for, what are some potential like warning signs that we're not the best fit, because someone explained to me finding a grad program and finding an advisor, you're having a relationship with this person. And if you know your style of learning is not the same as that person, it's not going to work. It might not be a reflection on either one of you being good or bad. It's just if you don't feel that they're going to foster your curiosity, push you to do work. For me, that was important. And so if I didn't sense that from someone else, if I sensed that they might leave me primarily on my own and not say hi or like not check in on me twice a year, I'd be okay. I'd survive. I've done it before, but it's not something I want for myself if I had the choice.
That does sound like a bummer.
Yeah.
Could I just jump in from an advisor perspective and say that what Jamie is saying is extremely wise? Some people want to be left alone, right? Some people want to have an independent project where their advisor only just makes sure they're still breathing once a year. And then some people need a different relationship. And so knowing that about yourself, I think is really important. And for students who are thinking of grad school, sometimes we look, at the news and we see someone that's very famous because they're doing the types of studies we want to be doing. And so while that is really exciting, you really want to make sure that that person has the time to give you the mentorship that you need. if you want that closer relationship. But if you want something that's very independent, then maybe that's fine to have an advisor who's, off trotting the world and giving talks every 5 minutes or so. So it really depends on what kind of mentorship you think you need as you're going into graduate school, and having that conversation with your advisor as well as the other trainees that are in the lab when you're applying to that lab or to that research group.
Totally.
Yes. I will also say networking is really important. I think it is really important. And I'm not saying that it gets you always in the door, but I think it's also like a way to kind of speed date your advisor. For me, to talk to them, to get to see, okay, who are they? Will that work? And then on the reverse, so then to see you, not just on a sheet of paper. And so I got connected to Maria through a previous advisor, Dr. Lorena Madrigal. And that's how I heard about Maria and the lab. I also want to say that I think I've been really grateful that we've been able to develop a relationship, Maria and I, that if there has been maybe some problems or discrepancies, I think we've been able to have moments of talking through that. And I don't think that can happen with every type of advisor. But I will say that probably one of the reasons why we're here today, and like she's pushing me to do these things and I'm receiving it well, and I'm trying to get out there and live up to what I have the ability to do, is because we have a sense of trust that we've been able to develop through conversations of, this isn't my style, this isn't like, what are we doing today, like I don't think that this is working, and then how can we change. And in reverse, I think I've done things that maybe didn't live up to expectation, and then have conversations about what we can do to improve that and things like that.
Thanks, Jamie. That's really sweet.
One thing I want to slide in and say is I think the word networking gets a bad rap. Like people are like, ugh, network. And it like feels so inauthentic and like gross and weird. But like networking it's not like you see in a movie from like the early 2000s with like business cards necessarily. Networking, it's also like grabbing coffee with someone, or just like saying, hi, I read your article, I really admired it. Here's like a question I had about it. Like, it doesn't have to be weird and painful. Like it is actually just having a normal conversation with another individual. That is what networking is.
Yeah, I agree. And also it allows you to identify a network of mentors, right? So Jamie said something about how she had different advisors throughout her career. I would say that no one person can give you all the mentorship that you need. And so the more people that you know within your field or even outside your field, you can identify that. So for example, maybe Jamie wants to do a really complicated phylogenetic analysis that I've never done, right? So then maybe I can connect her with someone that I know that has that specialty, and she can receive mentorship there, or vice versa. She already knows someone who knows how to do it and can talk to that person. Or if she wants to go into a career path that is not the path that I'm in, that's where that network is useful. And so who you know that can help you develop your potential is really important for graduate students, for undergraduate students as well, and even for postdoctoral fellows. A lot of the people that were my peers when I was a postdoc are now also assistant professors. And I know, hey, this person is an expert in X topic. I can send one of my students to that person. And that's literally how Jamie came to my lab. So those networks of collaboration and mentorship can be really essential to further your career in anthropology, but also beyond anthropology as well.
Oh, yeah. Yeah. Honestly, this is like a lesson that you can use for your entire life, because no one person can be all things to another person. This is also true for romantic relationships. Shout out Dr. Bowles, my therapist. But no, it is really helpful. I do want to be conscious of your time. So thank you for staying a little bit later to talk about this. We're going to wrap up the interview with the traditional 3 questions, which it's still 3 questions, even though there's 2 of you, but I guess asked twice. The first question is, if someone wants to learn more about DNA in general, or like the kind of work you do in particular, is there like a good introductory text, or is there a good place for them to start?
Yes. So because I'm so focused right now on ancient pathogens, the paper Hunting for Pathogens, Ancient DNA and the Historical Record by Stephanie Marciniak, 2016 is really good. And a really enjoyable book is called Plagues Upon the Earth, Disease and the Course of Human History by Kyle Harper. That's also really just, it's a big book, but it's like a really good story of how humans and diseases have played throughout all of human history.
Perfect. What about you, Doctor?
Yeah, I agree actually with those recommendations. There are a couple of different sort of introductory level texts that I could recommend. One, not to toot my own horn, that I recommend for undergraduates. Okay, I'll toot it a little bit. Is a chapter actually that I wrote when I was a student with my then advisor, Dr. Ann Stone, in a volume from 2018. It's called Biological Anthropology of the Human Skeleton. And our chapter is called Ancient DNA Analysis of Archaeological Remains. And it's a really introductory chapter that is meant to teach students kind of the basics that you need to know to then move on to more advanced genetic texts or bioarchaeological texts. And then if you want to read more about ethics in ancient DNA and sort of why we do things in this way, I can recommend a paper by Nieves-Colón and collaborators called Community Partnerships Are Fundamental to Ethical Ancient DNA Research. It was published in 2023 in the journal Human Genetics and Genomics Advances.
Perfect. I will make sure that those are all in the show notes. Don't worry. Second question, what are you reading for work right now?
I am reading a lot of papers about how to code and analyze DNA. It's very, so like a meta pipeline, Kraken unique.
Those are all bioinformatic tools that allow us to identify the presence of ancient microbes, and they're not simple to learn. And so reading a lot about kind of the baseline computational skills that you need to have has been a real big part of Jamie's training. And I think also my own training, right? Because it's not a standard piece of anthropology training. And I will just shout out a book that was really helpful for me. If anyone is thinking about these skills, it's called Bioinformatics Data Skills by Vince Buffalo. And it is such a great manual to like, how do you learn how to code? How do you learn to think like a coder? And then you can get to apply that to the questions that you're actually interested in.
Totally. I think probably once every other episode, I'm like, everyone should learn how to do math. And now I think I need to start adding everyone should learn how to code because it's like come up a few times, but it really can make a huge difference for you. Grad school won't suck nearly as much because those are always like those are always like the worst courses, like in your intro, like grad school statistics is the one that everyone hated. And then like intro to R, everyone was like, this is the worst thing I've ever done. But like, once you know, you're like, you feel so powerful.
I'm such a nerd. I totally loved intro to R.
That's how you know academia is for you. But yeah, I mean, a lot of it depends on the professor you have teaching it too. Cause like sometimes it's like, you're just like, all of my grad school level statistics, I taught myself out of a book and like this one YouTube channel. Like that's how I taught myself statistics. And I still use all of that all the time. All the time. Sorry. Okay. The last question for y'all is, what are you consuming for fun right now? So this can be like a TV show. It can be books. It can be, some people say, the great outdoors. What are you doing for fun?
I'm an inside person, so I for TV shows I really like Ted Lasso and Arcane. I play Dungeons and Dragons quite often, and that's a fun like way to have my imagination go wild. I also have been really into using my coding experience to apply it to things such as coding games, video games. So I use Godot and I make I'm trying to make little games and learning game development.
That's so cool.
Yeah.
What about you?
Well, I have a 1-year-old puppy who has a lot of energy. And so what I do in my free time is try to train my 1-year-old puppy, which I actually really enjoy. So she knows how to do quite a few tricks now. And then I like to read. So I like weird sort of magical realism and fictional stories. So, 100 Years of Solitude is a huge favorite of mine. So I like to read and play with my dog on my free time.
When I was in high school, all my friends called me Gabo because they all read 100 Years of Solitude at the same time, and that's the author's nickname. So I was going around with
I haven't seen the new Netflix series yet. I'm not quite ready for the intensity just yet. I need, a couple of days to binge it.
I mean, the book itself is really intense. I can't even imagine reading or watching it on film. I'll tell you guys what I'm reading right now because I do think it's like a really weird combination. I just finished Careless People, which is by, I think, Sarah Wynne Williams. She worked at Meta, which was Facebook, and she was on their global policy team. This whole book, it took me so long to read because I'd read like 10 pages and I'd have to put it down because I was like, I feel sick. It's nuts. I strongly recommend it to everyone, especially if you're thinking about working in tech. It feels very true. The other book that I'm reading right now is Thursday Murder Club by Richard Osman, which is like a murder mystery. And then I had a friend recommend this book to me. I'm going to go ahead and say it out loud because I don't think anyone should be ashamed of what they're reading. It's called The X-Hex, and it is so bad and it's so good. But if you're looking for, it's straight up a romance novel. It's just a romance novel. It is.
I'm currently reading this book. It's called Into the Forest by Christina Henry, and it's an anthology of female horror writers who write about the Baba Yaga. So it's like 20 stories. Each author has a different interpretation where the Baba Yaga is either the protagonist or you're in the world of the Baba Yaga, right? Who is this fictional sort of old witch character from like Slavic mythology. So it's very different from like, my own cultural background or even what I study, but it's kind of interesting. And it's an anthology. So, it's like, if you've ever watched Love, Death and Robots, right? Every short has a different kind of style. So this is the same idea, right? And it's kind of interesting. Some are short, some are long, and yeah.
Yeah, no, totally. I like to read like a lot of different things all at once. Honestly, the murder mystery and the romance novel at the same time, it's mostly because I just needed to like clear the taste of Careless People out of my mouth. Not because the writing was bad. It was excellent. I was just like so stressed out the entire time I was reading it. It took me literally a month. It never takes me that long to read a book. But yeah, anyway, that's actually it. Thank you both so much for joining. And I hope to hear from you all in the future.
Yeah, thank you so much for the invitation.
Yes, thank you for having us.
Thanks for joining me for this episode of the Anthrobiology Podcast. If you have questions or comments, you can reach me at gabby.lapera@anthrobiology.com. That's spelled GABY.LAPERA@anthrobiology.com. This show is produced by me. Music courtesy of Chad Crouch, aka Poddington Bear at Sound of Picture.